All right, let's get started. Again, thank you for everyone for joining us on our webinar. This is our third in our series on the techniques behind cell types knowledge explorer. And we have a great panelist today with Rachel, our scientist, Brian Lee, our senior scientist in our EPhys, and Stacy Sorensen, our associate director of neuroanatomy, will be representing our morphology team. Just a reminder, this webinar is recorded so if you miss it or want to rewatch it, it will be posted on our YouTube. It's also a series, so if you missed the other two webinars, they're all posted on our YouTube channel. We're happy to take questions. You can post the questions here on the chat, or if you are watching a recording or watching YouTube, feel free to put on the YouTube comments or our community forum for further questions. Today's agenda, so Rachel's going to start out with just going over the cell types knowledge explorer and how to access some of the Patch-Seq data there. Brian Lee will talk about the EPhys portion of Patch-Seq and kind of just also give a broad overview of what Patch-Seq is. Then we'll stop for 5 to 10 minutes for questions if anyone has questions about what they saw about the cell types knowledge explorer or EPhys. And then Stacy will talk about how morphology is measured with regards to Patch-Seq and why morphology is important. And with that, we'll kick it off to Rachel.
Hi, everyone. I'm Rachel Hostetler. If you joined us in the last couple webinars, I also presented in those. It's nice to see you again. Today, I'm going to give a brief introduction to the Cell Type Knowledge Explorer. And the Cell Type Knowledge Explorer is a tool that is used to access multimodal data from a mini atlas of the primary motor cortex. And all of these data came from a project that was led through the BICCN, which is the Brain Initiative Cell Census Network. And this was published in a series of 17 papers in 2021. And so the goal of this project was to create a mini atlas of the cells in the primary motor cortex. And so 3 species were chosen for this atlas. There were samples from human, from non-human primate, primarily from marmoset, and then also mouse. And a number of different techniques and experiments and studies were done on these samples, ranging from transcriptomic, single cell and single nucleus transcriptomic experiments, spatial transcriptomic experiments, Patch-Seq, multiple types of labeling experiments with transgenic lines or with different AAVs, and also single cell morphology. And all of these data combined resulted in the comprehensive characterization of cell types within the primary motor cortex of these 3 species, and over 3 million cells were profiled using these different modalities. And this resulted in about 200 terabytes of data, which was all made freely available through the Cell Type Knowledge Explorer, which I'll go over next. And again, I mentioned this paper, this study resulted in 17 papers. So hundreds of people worked on this project. It was a huge amount of data. And there's a summary flagship paper that was published in Nature in 2021. That's featured down here in the right. And yeah, this was a very large collaborative effort resulting, from hundreds of people across over 45 institutions. And this was all published in 2021. And so the main parts of this data that I would like to highlight are the taxonomies. So within this data, we had a transcriptomic taxonomy created from mouse. We also had a second taxonomy created from marmoset, and a third one from human. And these taxonomies were created specifically using single cell and single nucleus RNA-seq data, and these were all done individually here. If you want to read more about the mouse paper specifically, that's featured in Yao et al. If you want to read how these 3 data sets were created, all of them, you can read about that here in Bakken et al. And then these different taxonomies were then reintegrated to make one massive cohesive taxonomy. And in this taxonomy here, we can see where we differ between our 3 species and where we are more similar. So all the way on the left-hand side here, under LAMP 5.1, we can see that there's a 1-to-1 ratio between this cell type, between human, marmoset, and mouse. And then if you go further, perhaps looking at some of the p-val groups here in the middle, you can see that we have a lot of different cell types for all of the 3 different species. And so this is a good way to show where we're more similar between the species and where we're more different. And our next webinar on March 27th will specifically focus just on this cross-species aspect and how to reconcile the differences and work with data from different types of species, specifically transcriptomic data. And so all of this data, again, was generated in these studies, and then together with collaboration between the Allen Institute and other members of BICCN, the Cell Type Knowledge Explorer, or the CTKE, was created to access this data. So this data is online, freely available to use and you can access it through a web browser. And so in this next part, I'm going to show how you can actually access this data, how you can use the CTKE, and the type of information that you can get from it. And so this is on our website, brainmap.org, Atlases and Data, Cell Type Knowledge Explorer. Here I'm looking at the mouse data. Here we have a sunburst plot, which is showing the taxonomies of our different cells within the primary motor cortex. Here we're looking at the GABAergic subclasses. We have 7 subclasses here. Now we can look at the gene expression. We have the UMAPs available. We have a summary up here, which is showing us just looking at in our GABAergic cells. If we want to go further down in the taxonomy, we can click on a subset. So now we're specifically in the SST or somatostatin-like subclass. And then within the subclass, we had 19 different cell types. Again, we can look at the marker genes that were used to define the subclass. We can look at the UMAP here. And now we can start to look at our morphology data as well. So we can look at the different shapes of the 19 different cell types within the SST subclass. We can also compare between its siblings, how the SST cells look compared to other ones. We have electrophysiological data. So if you want to look at the amplitude of the action potentials, you want to look at the input resistance, what the traces look like. We also have some spatial transcriptomic data in this as well, and also some epigenetic data. And if you have any questions on how this data was generated, we have these little info bars here. So this is specifically a Tax-Seq data that is featured here in this epigenetic browser. And the more data buttons allow you to download the data or get access to it or look at a project summary to get a better understanding of how exactly that data was created. It's also linked to the references, so you can find the actual papers published. And then since this is a taxonomy, now we can go all the way down to our cell type level, so our most refined level here. And now we can see what are the genetic markers that sets this specific cell type apart from other cells within that subclass. So here we have more genetic information. We have more information on our morphologies and our reconstructions. We can now compare our electrophysiological data for these different cell types within the SST subclass, see what really sets these different cell types apart from each other. We can look at their example traces, look at their firing patterns, and we can also look at their homology. So since this was done on 3 different species, and these taxonomies were then integrated to create one taxonomy, we can look at homology. So here in dark red, we can see that there is a homologous cell that was identified in the human taxonomy. Here's an example. Here's what that cell looks like. Here's some information. Here are the genetic markers that were used in this human sample here. And then down again, back at our heat maps, we can then look at our third species, our marmoset. So we have mouse on the right, marmoset on the left. We can click on the darkest red one here, and that is the homologous cell type in the marmoset data. Here again, we can look at the different genetic markers that were used to define these cell types. And we can look at more information on what defines the cell type compared to the other cell types. And so that's just a very brief overview of the types of information that you can get from the Cell Type Knowledge Explorer. So it's all interactive, it's all free to use. If you just click through that sunburst plot that you saw in the beginning, those giant wheels, that'll take you through the different levels of the taxonomy. So you have your class level, subclass, and then cell type. And then at the cell type level, you can then look at homologies between your different species. And this is all, again, just in the primary motor cortex.
Hey, Rachel, we have a hand up for the Naparo if you want to ask your question.
Sure, yeah. Someone has a hand or
yes.
What? Sorry, I missed.
Yeah, thanks.
I can't see the thing.
Can you hear me?
Yep.
Yes.
So I have a couple of questions. So one is, when you say homology, is that just transcriptome based, or would that be sort of integrating functional and morphological data as well?
That is just transcriptome based.
Okay,
Sure.
and then
Yes.
yeah, so that, I guess that's the most useful, because then you'd be going to detailed functional studies or something. So then the other question, which is sort of immediately before that, when you show morphologies, and of course, so my question is how are you integrating multiple examples of that cell type? I mean, is this sort of averaged in some sort of image analysis software, or is this averaged across multiple examples of that type, or is this just one sort of key, sort of a standard type genus type of cell, or what?
Yeah, so for the morphologies, the traces that are actually shown, or the reconstructions that are shown in the cell type knowledge explorer, are actual cells that were traced. It's not like an average of, 10 cells. Those are example cells from individual cells. But the taxonomies themselves were based on the transcriptomic data in this experiment, and then yes, from that. So the morphologies were not used to create these specific taxonomies, but then, yeah, so that there are individual cells that are shown there instead of averaging.
And if I could quickly ask one last follow-up question. If you have, so this is all in one very small restricted part of the cortex, and I'm wondering if you, if this is really more from the research, your research question. If you saw a very similar transcriptome profile in a very different area, to what extent do you think the morphology would look similar?
Yeah, so I think because, primary motor cortex is a little unique because we don't have the typical layer 4 there like we do in other areas.
Right, and the projections are very, very different, and all of that. Yeah. Yeah,
so I do think that's something to keep in mind. I do know that so this was done in 2021, and then since then the Allen Institute and BICCN has done a whole mouse brain study where we have transcriptomic types across the entire mouse brain. That data does not include morphological data. That's just single cell, single nucleus, and spatial transcriptomic data. But I think in my experience, depending on specific cell types, like, inhibitory cell types tend to be more similar across cortical areas as opposed to excitatory cell types. You see more diversity in the excitatory than you do in inhibitory. So I think depending on, which layer you're looking in, if you're looking at layer 2, 3 across cortical cell types, that's going to be different than looking at layer 4, since we don't have a typical layer 4 in motor cortex. So I think, it's definitely cell type dependent. But these transcriptomic types we do know exist in other cortical areas outside of motor cortex. But I'm sure there's definitely variations, likely variations. I don't know how extensive those variations are, but in the morphology, especially if you're looking at layer 4 cell types.
And then
I know there's some
I'll
other
show some visual cortex and then motor cortex later on in the talk, so you can get a sense of those cross-cortical comparisons. But like there is a sort of like layer 4 like population in motor cortex as well. So
you took words out of my mouth, Stacy. We do have a whole morphology section, and so hopefully answer some of that questions on that one. Okay, great. Thank you.
Yes, Stacy will show some visual cortex data later in the talk. So great questions. So back to the cell type knowledge explorer, what are some potential use cases that you would have? Well, perhaps, you're studying a particular cell type in mouse, and you're interested if it has a transcriptomic homologous equivalent in human. This would give you a lead to that answer. Again, perhaps you're studying some cells in another cortical area, like we just mentioned. Maybe you're studying cells in somatosensory cortex or visual cortex, and you're curious how that compares to data in motor cortex. Or if you're studying motor cortex cells in a disease model, right? So all of the data here was shown from healthy, non-diseased control mice. So if you're studying a disease model and want a comparison, perhaps for your morphology, for your electrophysiology, for your transcriptomic data. Here's a control that you could compare to. If you want to look at the firing patterns or morphologies of specific genetically defined cells, this tool allows you to do that because we use Patch-Seq, and so we have that information on top of the transcriptomic information from individual cells. Also, if you're looking at layer-specific locations, these experiments were done in a layer-specific manner. And so if you want to look at differences between cells in layer 2, 3 versus layer 5, you have that information here, and you can break that down by layer specifics. And as I mentioned, Patch-Seq was used in these experiments to really combine the transcriptomic data with the morphological and electrophysiological. So here's an example, or here's a figure of what this transcriptomic taxonomy was made, how it looks like from the mouse. So this is purely just based on transcriptomic data. However, after doing Patch-Seq experiments and identifying certain electrophysiological properties, they were able to then connect and map on the electrophysiological properties and these labels here onto the taxonomy. And then the next step is with Patch-Seq, you're also able to look at the morphology of these cells through reconstructions from biocytin fills, right? So now we have this multimodal example of what these cells look like. And so how these experiments are done and how Patch-Seq works, we're going to go further into that with our next speaker, Brian, who is a senior scientist on the Patch-Seq team here at the Allen Institute.
All right, thank you, Rachel. Okay, so let's see. Yeah, so like Rachel said, my name is Brian. I'm a senior scientist in the Integrative Cell Physiology group here at the Allen Institute. And I have a team of patchers where we conduct Patch-Seq experiments. So today I'm going to take a few minutes to chat about what that technique is and how we can look at some of that data and understand what's actually in the CTKE. So here at the Allen Institute, we use a variety of techniques to understand the cell type properties and how to classify cell types. So this image is a nice depiction of how we have various modalities that are used to identify a cell type. So for transcriptomics, we have single cell or single nucleus RNA-seq. We have anatomy and morphology, then you have electrophysiology, connectivity, uses full morphology and synaptic physiology with the EM techniques. But really, we want to figure out what is the best way to classify a cell type. And each of one of these independent modalities are nice, but we want to understand what is the correspondence from one another. How does transcriptomics relate to connectivity or electrophysiology or morphology? And that's what Patch-Seq allows us to do. It allows us to bridge and make that correspondence to how each of these independent modalities relate to one another. So what is Patch-Seq? So Patch-Seq is a deviation from the patch clamp technique, which was developed back in the 70s by Erwin Nair and Bert Sackman. And essentially what the patch clamp technique is, you take a small glass pipette or electrode, and you lower that down to the surface or somatic membrane of a living cell, and you apply a small amount of negative pressure, and you rupture the somatic membrane that's under the tip of that pipette. And what that does is that gives you electrical access to the neuron, where then you can probe that neuron for the functional characteristics of that neuron. So what does a firing pattern look like? What is the resting membrane potential? And then a few years later, several other scientists and groups found out that with this internal solution that we are measuring the electrical activity of the neuron, we can actually put a dye into that neuron and understand what the shape or the morphology of that neuron is. So this technique, like I said, it's been around since the 70s and has been a great technique to understand how cells functions and what are the shapes of the neuron. But where we've come into play and what the purpose of this talk is looking at Patch-Seq. And this is essentially an adaptation of that technique that was developed by others and then optimized by the Allen Institute over the past few years. And what this technique is instead of just taking that pipette off of the cell and throwing it away when the experiment is done, we actually apply a small amount of negative pressure and extract the cellular components of that neuron to send that on for single cell RNA sequencing. So this is really a way to get 3 independent data modalities from one single cell and really start to make the correspondence for how these modalities are related to one another. So what is Patch-Seq and how does it actually work? So here's a depiction of a few cells. And as you can see from the left-hand side, we have a glass pipette where we patch onto the neuron. And you can see that the blue internal solution diffuses throughout the neuron, and that contains the dye that allows us to fill the cell and ultimately obtain the morphological anatomy of that neuron. During the same time, we apply a series of current steps to understand what the voltage properties or the intrinsic membrane properties of the neuron are. And then once that is complete, we apply a small amount of negative pressure where we extract some of the cytosol as well as the nucleus. And so embedded within that is the RNA from that neuron. And this is how we're able to do the single cell RNA sequencing from this particular neuron. And what I want to show here, and what's really important to obtain this, what we call triple modality data, is we need to keep the cell intact to a point where that cell can maintain the dye that we had just filled the neuron with. So what you can see is that the somatic membrane is stretching as we retract the pipette. And then ultimately, that somatic membrane will snap and reseal around the nucleus, but more importantly, will snap and reseal around the soma of that cell, holding in that dye that allows us to visualize the dye later on for morphological analysis. So this technique gives us the transcriptome, RNA-seq, morphology, and electrophysiology from one single neuron, thus, as of now, giving us really the best way to understand what is the true identity or what makes up the properties of a single neuron. Here is a video that we have taken from one of our rigs. You can see the pipette as we have applied the amount of negative pressure, and you can see that pulling the nucleus through the tissue and ultimately out of the brain slice. And so what was surprising to us at first is the nucleus is actually quite big. So this is where it's important to do this in a slow and methodical process because, like I said, we want to preserve the somatic membrane. Okay, so here at the Allen Institute, we do things at scale. We collect a lot of data that allows us to share this data, but in order to do that, we have to do it in a standardized manner. So here's the depiction of the Patch-Seq, and what I want to show right here is one of the features that we have to help collect standardized data is that we have our electrophysiology acquisition platform, which we call MEES, and that's available through our website. But what this does is this allows us to conduct a quick experiment to collect the most cells that we can in a day. But in order to do things quick, we also want to make sure that the data is of the highest quality. So ongoing during this experiment, QC metrics are being evaluated and applied and letting the users know if this cell is meeting the standards for what we have initially designed and set up. So it's a nice way to ensure that the data that we're collecting is of the utmost quality. Let's see. And then also importantly is that we have a series of standardized stimulus sets that we can apply to the cell across various regions and species. So the goal for these stimulus sets are to assess a diverse array of electrophysiological features, but still enabling a good and equal comparison across cells. So we have 5 main stimulus sets that we do. On the left-hand panel or left-hand column, we have a subthreshold, a Rio base, a suprathreshold in the long and short square format, and then the ramp. And then on the right is just an example of voltage traces of how cells and their phenotypic responses to these current stimulus sets. So navigating through the CTKE, how do we apply and look at this Patch-Seq data using this platform? So Rachel had a nice description of what's embedded within this platform. So I want to revisit this a bit. So in the middle is that sunburst plot that she had shown. And on the left and right-hand side, we have the gene expression UMAP, essentially the genes that the neuron expresses for the broader class of neurons. So the orange are the GABAergic and the blue are the glutamatergic. And then I've colored over the non-neuronal types because that is not of interest for Patch-Seq experiments at the time. And then taking that a step further, as Rachel explained, each embedded within each one of these broader classes are subclasses. So for the GABAergic type, we have LAM5, VIP, SST, P-valve, and then several other glutamatergic types. So how does this apply to physiology? So within each one of these subclasses, I have some representative voltage traces from each of these subclasses, and you can see that these are voltage responses to a hyperpolarizing pulse as well as a superthreshold pulse. So you can see the train of action potentials to a suprathreshold response. And then on the right-hand side, we have representative examples from the GABAergic subclasses. So how can we look at these and start to identify how cells may differ or how cells are similar? So I have the same traces on the previous window or the previous slide and sorted by glutamatergic and GABAergic. And hopefully what you can appreciate right off the bat is that GABAergic are actually look quite different compared to the glutamatergic. The glutamatergic are qualitatively look pretty similar. There's a few features that we can use to identify these cells. We have what's called adapting, and you can see from this example that the cell actually fires pretty quickly, but then slows down over the course of the stimulus. We have bursting-like cells where we have a group of action potentials that are initiated in the beginning, and then we have what we consider normal or regular spiking after that burst-like period. But for GABAergic, they're actually more heterogeneous and diverse in terms of their properties. We still have some adapting spiking properties. We have irregular, we have bursts, but the bursts are larger bouts of action potentials compared to a glutamatergic neuron. And I think what's most well known across the community are fast spiking parvalbumin and neurons. So that's obviously, you can't even see the number of action potentials. It looks just like a block, but there are indeed a high number of action potentials that are embedded within here. So this is a qualitative look at the firing properties. And what we can do next is that we can look at a single action potential and see how that, what the features of that are across the different subclasses. So again, the same plot, a similar setup as what I showed before, except for this time I'm looking at a single action potential. So this is a very zoomed in version. And you can see what we call the kinetics of an action potential. How fast does it rise? How fast does it drop? And then what is the AHP, the after hyperpolarization, or the peak? What does that look like? And really looking at these one by one is hard to tell, but when you bring them in and normalize them to the peak, then you can really start to see how they are different. So on the bottom, we have the glutamatergic types, and really there's not much difference in terms of the rise and the drop of the action potential. There's quite a bit of diversity across the after hyperpolarization, but within the GABAergic types, that's where we see more diversity. So this one on the inside is a parvalbumin, so the fast spiker, so that has faster kinetics, so meaning that it rises faster and drops faster, and this is what is responsible for the fast spiking like properties that we saw on the previous slide. So those were qualitative looks at the firing properties and the action potential, but we want to take a quantitative approach. How can we quantitate some of these features? So on the left-hand side, I have a depiction of the stimulus sets that I showed before. And then with the Institute, we have this package that's called the Intrinsic Physiological Feature Extraction, or IPFX. So this is a package that's available through our Institute. And what this does is this takes our data set and will automatically extract features from that particular experiment. And there's something like 50 plus features. But what I want to do right now is just highlight a few features that can really help you distinguish different types of neurons. So the resting membrane potential is one of them, looking at not only across all of the properties, but when we have the subthreshold properties, we can look at the degree of sag, and that's mediated by HDN channels and can be different across different cell types. We have the firing rate, which is essentially the number of action potentials per second or per 100 milliseconds. We can look at the interspike intervals, how fast, what is the time difference between this first spike and last spike? You know, this will tell us if the cell is accommodating or has burst-like properties. But I think that the most important one that can help distinguish between certain types, especially GABAergic type, are the kinetics of an action potential. So essentially the upstroke, the downstroke, how wide is that action potential, the peak, and then the after hyperpolarization. So all these features are automatically obtained with each recording. A lot of these features are listed within the CTKE, as Rachel showed. And I think really the most powerful thing that we can do with this Patch-Seq is really start to explore the function and gene relationship between these experiments. So here I have just some representative examples of traces, but since this is Patch-Seq data, we're collecting on average 8,000 genes from each of these individual experiments. So we can start to look at, okay, here is a cell that may have diverse electrophysiological properties, and what are the genes that could be responsible for that? For example, we have this single action potential right here. We know that sodium is responsible for the upstroke, and we know that potassium is responsible for the downstroke. What different subunits are involved with a particular cell? We have a fast spiking-like property. Are there different subunits that are responsible? We know that they are from previous studies looking at KV 3.1, but there has really never been any direct relationship to looking at the KV 3.1 gene. So this is where Patch-Seq really allows us to get into the finer details of how genes influence function of a neuron. And so just to kind of summarize, Patch-Seq is a powerful experiment that allows us to obtain multimodal data from a single neuron. I went over the electrophysiology and transcriptomics from these experiments. And so on the middle, I have an example of one of our stellar stellar examples where we had 10 neurons from a human slice. And, you know, with these two modalities, we can get the gene expression, we get to electrophysiology, but we can't forget about the third component, and that is morphology. And that is where we can look at the shape of that neuron and really get a better view of what is going on with this triple modality data, and how can we really begin to classify these neurons. So with that, Stacy will be talking next about morphology and how we use this to classify and characterize neurons.
But before we go to Stacy with morphology, if anyone has some questions they would like to put in the chat, either the Zoom chat or the YouTube chat, feel free to put them there. We already got one question. Great presentation. Have you tried to plot the action potential on delta V/delta T versus the membrane potential plot?
Yes, we do that on a routine basis. That's part of our core set of analyses, and that's been plotted in several of our papers. One is Stacy's paper that's on bioarchives, as well as a paper in Cells, which is our first Patch-Seq paper, and those delta plots are all on there for each of the action potentials and a summary for the specific transcriptomic types.
Great, thank you. If anyone else has a couple questions, we'll wait a minute. Otherwise, we also have a Q&A after Stacy. So if there's a question that comes up, feel free to chat it, and Brian will still be here to help answer that. But since we don't have any questions, I'll let Stacy take it over.
Thanks, Lauren. Great job, Brian. Right. It's a little bit different than what it looked like before for me, but I'll try this. Yeah. That's not what I want to do. Hold on. I'm not, maybe, I'm sure why I'm not getting the right file showing up, even though
I can also share your screen for you, Stacy, and slide control if you would like to do that.
Sure, that sounds good. Okay,
All right, should be set up.
go ahead and advance to the next slide. Thank you. So the existence of diverse cell types has been known for more than 100 years, since the first descriptions of cell morphology created by Santiago Ramon y Cajal. And an example of one of his iconic drawings that displays this is over here on the right. So since then, a rich literature has evolved surrounding morphological cell types, and some examples of that are also shown here below for cortical inhibitory neurons on the left, and bipolar retinal neurons on the right. And then, as you have been hearing, morphology continues to be a stable and valuable indicator of cell types. And with the patch clamp and Patch-Seq work, as well as lots of other stuff, we are now starting to understand the relationship across cellular properties for many different cell types. If you go to the next slide. So one of the important the things that we get excited about with morphology is as a component of defining cell types is that the distribution of dendritic and axonal branches, and also the anatomical location of the soma, give us clues about which neurons they exchange information with and the functional role that they might play. So here I'm showing an example from a review paper where neurons in layer 4, you cannot see my cursor probably, no. But neurons in layer 4 of the visual cortex, for example, they receive inputs from the visual thalamic nucleus, LG, and then they send information to neurons in layer 2, 3 and layer 5 that either send information to brain regions involved in sensory processing, such as higher order cortical areas like V2, or to subcortical brain regions that are involved in generating a motor output. So just having an understanding of how morphologies are distributed with respect to these compartments that we understand at a functional level gives us a lot of important clues about what the cells might be doing in the circuit. If you go to the next slide, that would be great. So to access morphological and anatomical information about Patch-Seq neurons, we have to fix the slices that Brian's team has recorded from and filled, and then we stain the slices and image them at different resolutions. So here I'm showing that sort of gold standard slice that Brian showed, but without those the reconstructions, the multicolored reconstructions on top of them. If you advance again, Lauren, you can see that we take a 20x overview image of the slice, but then even at this lower resolution, we can still get a sense of the overall morphology of the neurons. But if you go to the next slide, that slice overview image is also really important for our pipeline. So we use it both to assign brain region and to get some quantitative information about layers. So the way that we get brain region is that we align the 20x slice to our CCF, or Common Coordinate Framework. This is an anatomical reference atlas that we can use to understand where our neurons are in the brain. And then in addition, to be able to better see the layers, we also stain all of our slices with DAPI, which is a nuclear stain. And we take a second 20x slice image. Here I'm showing a zoom up view of one of those images in the middle. DAPI helps us to see the changes in cell density that indicate which layers a cell is in or its branches are in. And then we can draw the borders between the layers which we can then use to measure morphological properties relative to those different functional compartments. And then finally, important for morphology is that individual neurons are also imaged in 3D at 63x to get a higher resolution image for reconstructions. And then we use freely available VAW3D software to reconstruct our neurons. If you go to the next slide, that'd be great. Okay, let's see. So to extract quantitative features from morphologies across a large data set of different types of neurons, we save our reconstructions in the SWC file format. We make sure that all of our SWC files have been processed in the exact same way so that there are no, formatting issues in comparing across neurons. And then we also make sure that these SWC files are compatible with neuron modeling software, which has specific requirements for parent-child relationships and that kind of thing in a tree structure. And then just a little more information about how we get the slices into the CCF. We use fiducials to align them. And then using the layer drawings, we also upright our cortical neurons and then place them in an average cortical framework shown here on the right, so that we can compare the layer distribution patterns across neurons. This gets a little funky when we have motor cortex, for example, where we don't have a layer 4, and that's something that we're still kind of figuring out how to deal with. But that would, when we do that, we have the average cortical framework that doesn't have a layer 4. So but there are some subtleties there that we're still figuring out for comparing across brain regions. So to calculate morphological features like total axon length or the number of branches or even, surface area of the dendrites, we run each SWC file through a skeleton keys package, which is publicly available online. And then we also calculate these branch distribution histograms, which report the amount of dendritic or axonal length at 5-micron intervals throughout the cortical depth. And these features can then be used to quantitatively compare morphologies across transcriptomic types and then relative to other cellular properties like EFIS. And then these branch histograms appear on our CTKE site, and as you'll see, are pretty important to us. So looking at back at the transcriptomic taxonomy on the CTKE, we'll take a look at morphologies across the transcriptomic space. So glutamatergic neurons, which appear down at the bottom in cool colors, which to kind of indicate that they activate other neurons, tell other neurons to go. And then GABAergic neurons over there on the right, which appear in warm colors to indicate stop, since these neurons inhibit other neurons. The non-neuronal cells, as Brian mentioned, we're not really focused on for these particular studies, but they appear in more neutral colors. And so we currently don't have any morphologies from glial cells, but could be in our future, who knows? So we'll start with looking at glutamatergic pyramidal neurons, since most people are familiar with those. So thanks, Lauren. So through the lens of transcriptomic types versus the morphological types that I have mentioned previously, we also find morphological heterogeneity in glutamatergic neurons. So as you can see, different types of pyramidal neurons are found in different layers. The layer markers are indicated on the right of the histograms, and then the amount of branch length is then down here on the x-axis. And then this is an average. Well, in this case, we're showing the branch histogram for just this particular neuron, but when we actually analyze the data, it will generate an average histogram. So layer 2-3 neurons, or layer 2-3 IT or intratelencephalic projecting neurons, mainly have dendritic branches in layer 1 through layer 2-3, while these layer 4-5 IT neurons, the 4-5 meant to capture this difference in the presence or absence of layer 4 and 5 across cortical brain regions. But they have, if you'll notice, this is a neuron from motor cortex, but it looks very similar to a untufted or star pyramidal layer 4 neuron that you would find in, somatosensory cortex or any other granular cortex. So, and then over here on the right, we're showing a layer 5 ET or extra telencephalic projecting neuron, which has many dendrites, mainly distributed in that upper part of layer 5, though these neurons are found throughout the depth of layer 5 in this cortical brain region. If moving on, so similarly, GABAergic interneurons, even within the same molecular subclass, in this case, somatostatin or SST, can have very different soma distributions and morphologies. So SST CalB4 there on the left have axon primarily located in layer 1, which is a defining feature of Martinotti neurons, and also down in layer 2-3, while the SST, we call it HIPSE, but HPSE neurons have cell bodies and much of their local axon located at the border between layer 2-3 and layer 5, and then have a very minimal axon up in layer 1. So though these are closely related types transcriptomically, the axon distribution patterns can be very different, indicating that these neurons are likely to contact different cells. Later on in the talk, I'll give you a hint about that is in fact the case. If you move on to the next slide, thank you very much. So when we look across the entire GABAergic landscape, we see that morphologies are frequently similar within a type and then different across type, which gives support to the validity of these transcriptomically defined types. And I just want to point out that I'm now showing our visual cortical data set. And if you look down at some of these SST neurons that are here in yellow and orange, and you'll see that they have some similar shapes to the motor cortex interneurons that we looked at, like Rachel mentioned. Okay, so if we now go to the next slide. So if we then combine morphology, electrophysiology, and transcriptomic data from these Patch-Seq experiments, we can generate taxonomies of multimodal MET types, which we've now done for GABAergic and glutamatergic neurons in mouse visual cortex. And we can also I just want to point out that you can find the GABAergic MET types for visual cortex also available on our website, though in a different location that maybe somebody can share in the chat. Moving on to the next slide. So we're also using morphology in a similar manner to how transcriptomic data is used as a Rosetta Stone, or as a way to link neurons across anatomical data sets to extend our multimodal cell type definitions to include synaptic level connectivity, like here on the left, as I'm showing for a subset of SST MET types, where we do see different connectivity profiles for the different morphological shapes that we've identified with this approach. And then also on the right, we've done this for glutamatergic neurons in visual cortex by bringing in long-range axonal projections that we aren't able to access in our slice preparations. And I'm showing those data here for the glutamatergic MET type taxonomy. And so with that sort of teaser, I'll end there and be happy to take any questions.
Big questions, and also, let's see. Also, I just launched a poll. If you're on the Zoom, if you have any feedback, just a quick one question, basically, if this webinar series helps, these polls will really help us with figuring out who's in the audience, and does this help with you understanding cell types or Patch-Seq? And if you're on YouTube, the link is on SurveyMonkey and will be underneath. And if you're watching a recording, it should be still in the YouTube description. And with that, if anyone has questions, either about Brian, about the morphology, or EFIS or Patch-Seq, let us know. Yep. Let's go panelist. Yeah, feel free. I see Nafuro, you have your hand up. Feel free to unmute.
Oh yeah, I'm just looking forward to seeing when you guys have information on sort of comparing, contrasting, whatever, from different brain areas, because I'm obviously the connectivities of the cells are so very different, and I think that that's got to be reflected, but I don't know, maybe some of the functional features might be similar enough that that's reflected in the transcriptomic. So I'm really looking forward to a more integrated view across brain regions. I know that's, you know, it's obvious and it'll be happening.
Yeah, we are kind of we've been moving down into subcortical brain regions and maybe less so focused on going across cortical brain regions, but I'm definitely very interested in that question as well. I think like maybe Brian wants to say a little something about in our human data set, we're collecting Patch-Seq data sets across different cortical brain regions, and I know that there is a plan to analyze that cross areal differences.
Yeah, it's definitely a really big interest of ours. And Stacey said that we also do Patch-Seq experiments in human and non-human primates. So making those cross areal comparisons is one of our top priorities right now. And human data and monkey data, as you can imagine, comes in a little bit slower. But some of the early results suggest, as you would expect, especially with the excitatory or glutamatergic types, that there are differences in physiology and morphology for certain types. So I think we just need to allow our databases and data sets to get a little bit bigger to make some really definitive conclusions. But it is a top priority of ours.
And just to plug, if you're more interested about the cross species, next webinar in March, we'll have some talking about, you know, how do you reconcile cross species and connect them. If there is no more questions in the chat, I'm happy to give people 5 minutes back in their day. Thank you, Stacey and Brian, Rachel, for this great webinar series. And thank you for everyone who joined us.
Thank
Thanks
you
so much for listening.
Thank you.